for women. So if we say on average a human's blood volume is around 5 liters, then we would be accurate. So the pH of blood, here we see the pH of blood is 7.35 to 7.45. And I want to talk about that for just, all right, that's a little bit better. And let's go ahead and think about that. So we said that we have pH, and if we have 1 and we have 14, oops, let's put 14 here, and then we have 1 down here, right? And then we have blood in the middle, 7.35 to 7.45. Now, if you go up the scale in that, let's say you have a higher number, like let's say this is 8 and 9, and we're going up the scale. What's actually happening is you're going down in your hydrogen ion concentration, right? So this is more alkaline. So a higher number is going to be more alkaline. Whereas if you're going this way, you know, like stomach acid and battery acid are real low down here on this chart. So we know stomach acid is, you know, down around 1. So as we're going this way, we're actually increasing our hydrogen ion concentration. So what that means is if I had a pH number, and let's say that I had 7.8, all right? And if we had this, you know, this number, this would be more alkaline in terms of what blood is concerned, versus if I had a pH of 6, this would be more acidic. And remember, we're talking about blood. All right, so this is going to be our line that we want to consider in terms of what we need to stay between. If we should go outside of this scale too much, so let me go ahead and clear this, and let's say that we have it more here, and we have 7.35 to 7.45 here. If we go this way too far, this can lead to death. And if we go this way, it can also lead to death. So pH balance for the blood between these numbers is essential. And we're going to see that one of the duties of the blood is to regulate the pH of the body. It needs to stay within this range. Does that make sense? Okay, so let's go ahead up here and let's clear this screen and over here, and hopefully we'll easily get back to our PowerPoint. Okay. Okay. So when we look at the characteristics here of blood, and we go back to our slideshow here, right? So let's go from, we see that we have that balance there, 7.35 to 7.45. We know that blood is viscous and a little bit sticky, all right? So it has a viscosity that is about 5 times greater than water. So when you're thinking about the viscosity of something, you're thinking of the thickness. So we know blood is thicker than water, right? That's in a song. Okay. All right. So blood has some basic functions and some really complicated functions. So I'm going to focus here on this very first statement, where we see that with this very first statement, we see here that blood transports oxygen, carbon dioxide, nutrients, hormones, heat, weight, and waste products. All right, and we just talked a little bit about this pH, so we know that blood also has the role of regulating. And then a little bit later, we're going to see that blood protects the body. So let's look here. We're going to start right here with this very first 3 words. Blood transports oxygen. So we want to know, how does that actually happen? And in order to show you that, we're going to go back to the drawing board here. All right, and we're going to clear and share. Okay. So remember what I just did. So what I did was, oops, clear. I showed you that vacutainer, and we can kind of still see it on the side a little bit there. All right. So here's this vacutainer again, right? And we said that when we spin the blood, it separates out into its component parts. And we said that we have red blood cells, white blood cells, and platelets, which are our formed elements. And then we have the plasma here, right? So let's go ahead and write these in. So we have the plasma, we have the Buffy coat, and hopefully you guys at home are writing along with me, all right? And then we have the red blood cells. Now, if you notice here, red blood cells make up a very big portion of this blood that we have just spun. So we say that the formed elements here, and remember, living in this Buffy coat, we had white blood cells and platelets. So the formed elements, we know that they make up 45% of whole blood, and then the plasma is going to make up 55%. So together, that equals 100%. But let's look here, all right? If we have this red blood cells, and I'm going to go ahead and clear this screen here again, all right? We're going to, remember I said things have a couple different names. So let's go ahead and look. Really, that Buffy coat is just a very fine layer on top here. So when we have the packed red blood cell volume, we have the red blood cells. We're going to give them a second name, and they are medically known as erythrocytes, right? So the packed red blood cell volume, this is very important, is known as hematocrit. So once we spin the blood and we get this packed red blood cell volume, we say that that packed red blood cell volume of erythrocytes is known as hematocrit, right? And we know that, you know, since this is a very fine line, it's going to be right around 45%. So let's think about these red blood cells. Let's think about what they're important for, all right? So the red blood cells, they kind of look like this. They look like a biconcave disc, all right? And the reason they look this way is because they have no nucleus. The nucleus was ejected during development, right? And inside of these red blood cells, what we see is we see these proteins, these iron-containing proteins, and there are millions of them, all right? So let's go ahead and let's give them a name. So we know those to be hemoglobin, right? And let's say that this is, we said that this is an iron containing protein. All right. So to put this into perspective, we know that we have our hematocrit. We're going to spin the blood, right? So this is the packed red blood cell volume. So we have the hematocrit. And when we take a look at these red blood cells individually, we see that they are just packed with these iron-containing proteins, which we know to be hemoglobin. So each hemoglobin molecule is very interesting because it's actually got 4 globular proteins. So if we think about it like this, right, this is one hemoglobin. So it's got 4 globular proteins. And remember, there are millions, and this is how it's abbreviated. Centrally located to the hemoglobin is an iron component, right? So we know that they are iron containing. Your body doesn't make iron. It does recycle it a little bit, but we have to consume it through our diet. And the reason iron is so very, very important is because iron is the part that's going to carry that oxygen molecule. So in that statement back there, we said that blood carries oxygen. So if we think about this, we need the iron to attach to the oxygen, and this is how most of the oxygen moves through our body within the hemoglobin. So if we have low iron, right, that can affect the what? That can affect the oxygen. Something very interesting that I had found was if you think about this, so my background is actually in, when I came to Deltec many years ago, I had first worked at Christiana Hospital in the clinical nutrition department. And something that is important when we think about iron containing protein, we always hear, we're not supposed to allow children to have exposure to lead. So with lead, we want no exposure, right? And the reason why lead has been taken out of pipes or has taken out of pencils and paint and things like that, and we want to watch for it in our plates so that it's not in our water. Well, why? Why is this a problem? And the reason is because lead, this affects iron absorption, all right? So lead, I'm going to say lead hurts iron absorption in the body, all right? So if you have low iron, right, that also affects, all right, remember this is going to affect the oxygen carrying capacity. So, there's other things that lead is going to also affect. Some of the minerals. Iron is a mineral that, calcium and magnesium and some important nutrients for brain function. But if it's affecting it here, what is that going to happen? What's going to happen in a developing brain? I want you to think about it and consider a developing brain, all right? So needs lots of nutrients, but here, this is going to be an issue if it, if it happens during childhood even more. So this is the big thing that you don't read about. We hear all these things that lead affects the brain, it affects neurodevelopment, and this is one of the reasons why is because it affects those major minerals, especially in children during development. Is there any questions there? No? Okay. All right, so let me go ahead and click out of this. Okay, and we're going to go back to our PowerPoint here. Okay, so we know now when we look at this here, we said that blood transports oxygen. And we know that it transports oxygen on those red blood cells. So remember, the red blood cells have hemoglobin, and that hemoglobin molecule is an iron containing, oops, I keep skipping my letters there, containing protein. and you know that the iron containing protein carries oxygen, right? So that is the big deal with red blood cells. Now, what we're also going to see later is that these red blood cells, when they distribute the oxygen, they're also going to be able to pick up some carbon dioxide. And we'll talk about how the blood does that when we get to the respiratory system a little bit more. Nutrients and hormones and heat and waste products, these things are going to all be carried throughout the blood. But what happens here is we see that that is the job here of the plasma, right? So plasma, we said, is 91.5%, right? But what we see in the plasma is that we see our nutrients, we see our hormones, we see waste products, and this is where we also can carry some heat. All right, so let's go ahead and click out of this, clear all drawings, click here, and let's move forward. Okay, so this here, this is what I was drawing for you, and this is that vacutainer. So a couple key things on here you want to remember that we just went over. All right, we said that blood is connective tissue. All right, and remember why it's connective tissue, because it has a matrix. So we said that the plasma is the matrix. All right, and we said these red blood cells, also known as erythrocytes, we have that packed red blood cell volume. All right. All right, so let's go on a little bit further here. And this next slide is, showing you the breakdown of the individual components. So we're gonna come back to this in just a minute, right? And what I would like to do now is I'd like to have some polling questions for you. All right. See if this is going to work. All right. Okay. So what are the three main functions of blood? So I'm going to go ahead and launch that poll. So what are the three main functions of blood? Yeah, 2 more seconds there. Okay. All right. So the three main functions of blood is that it transports, it regulates, and it protects. So all of the above would have been correct for that. All right. Right. Let's go ahead and go to the next one here. So we have all of the above, right? And go on to the next one here, right? The formed elements are red blood cells, white blood cells, and platelets. Let me go ahead and launch that. Is that true or is that false? All right. Okay, so that is true. Okay, next one. What percentage of whole blood is plasma? All right. So the percentage that is plasma is 55%. So 55% of whole blood is plasma. Right? Next one. Okay. Water makes up what percentage of plasma? Okay. All right. So 91.5% was correct on that. So that was the answer there. All right. And we just have one more here. Right. So the buffy coat in centrifuge blood is made of leukocytes and what else? So I have three that didn't answer yet. Okay, so the answer to this question is thrombocytes. So thrombocytes are also known as platelets, right? And I lied, I think there's one more. There we go. All right, so nutrients, hormones, and waste are all found in what? One more. All right, so nutrients, hormones, and waste are all found inside of the plasma. Okay, so let's think about that for a minute, right? When we look at this here, right, and we look at the Vacutainer here, we want to know that the formed elements, right, are white blood cells and platelets. And I did throw that in there with the thrombocytes. So platelets are also known as thrombocytes. So they are known as clotting cells, so thrombocytes. And they live right here. They live right here in this buffy coat. So this is what is contained here. And then the red blood cells, we have the packed red blood cell volume. Everything else is going to be up here. So we have nutrients such as glucose and proteins and things like that, hormones, all are going to live and inside of this, and waste products all going to live inside of the plasma, and even some gases, all right, such as CO2. All right, so we have in the red blood cells, we're going to carry most of our oxygen. things like that. But mostly nutrients, hormones, waste products, and so on are going to be part of that plasma. All right, so let's go ahead and move on to the next step here. I know it's getting late, huh? All right. So when we think about these blood cells, we want to think about where they're formed. And if you look up here, we have the main cell, which is a stem cell. So we have, this is going to develop into two parts of stem cells. So we have myeloid cells and we had lymphoid cells. So this main stem cell here is going to be the major part of where all of our blood cells are going to come from. And we'll go a little bit more specific in a later video. So what I would like to do with you is I'd like to talk about, this very first cell here. And as you see, we have what we call a reticulocyte. Now, when red blood cells are developed, that nucleus, as we said, is ejected, and it gives us a reticulocyte. And these are an immature cell. They will last about 1 to 2 days until the red blood cell is formed. Right?
It kind of looks like a sickle cell.
It does. It does look a little bit like sickle cell, doesn't it?
Yeah.
So when we think about the red blood cells, we want to think about hemoglobin, and we want to think about how important iron is, and then kind of, what it looks like. And then we want to consider, if it's so important, it's carrying the oxygen to our nervous system. I mean, it's oxygen to your body is almost like electricity to your house. So it's so important. So these red blood cells have such a major job. So let's think about this for a minute. When we look at these red blood cells, we want to consider what needs to fall in order for the body to develop more. And the answer to that is hypoxia, right? So hypoxia actually means low oxygen levels. And for healthy individuals, you're not even going to feel this. Like your body's just going to continue to make red blood cells. So red blood cells, they live about 120 days. So this is the life of a red blood cell. So they live and they die. And when they die, we have organs that are going to take care of them, such as the liver and the spleen, and, break them down. But when the oxygen dips a little bit, what happens is that we have the kidneys. So let's write this down here. We have low oxygen, which I'm going to write this again, we know to be hypoxia, right? And what this does is it stimulates the kidneys. So this is a kidney, right? So the kidneys to release a hormone called EPO. And the big name for that is erythropoietin, all right? So we call that in medicine, we say EPO. So erythropoietin, what that does is it stimulates red marrow, and this is one of the places where we find red marrow in our long bones. All right, so the process is called erythropoiesis. All right, so erythropoiesis, poiesis in itself means formation, and erythro means red. So when we read medical terms, we always read them from the end of the word to the beginning. So what this word actually means is formation of red. So formation of red cells. So this is a key component. Your oxygen level dips a little bit, kidneys release EPO, and then your red marrow is going to start to make red blood cells. And remember, what happens is that we have that stem cell, so part of the myeloid part there, it's going to eventually release what we call a reticulocyte, right? And then this is going to become an erythrocyte. So we have, then we have this more mature red blood cells. And as long as we're taking care of having all the components that we need in our diet, then we should, if we are healthy, going to produce normal red blood cells. So what other things are going to be important in red blood cells? So some of the things that that we want to just mention real quick while I have you here, all right? So nutrients that are important are, we have B12, we have iron, we have folic acid, right? And then of course we have protein. So all of these are important in red blood cells. So a deficiency in any one of these can affect the capacity or the job of the red blood cells. So these are, specific nutrients, and we have to make sure that we eat them all. So they're all important to obtain every day, especially during the growing years. So a deficiency of any one of those nutrients would not be a good thing.
Does that mean why they put women on the big horse pills, the iron pills and stuff to help, because they have to gain more blood volume
So when you mean when a
when
woman,
they're pregnant?
yeah, so they shouldn't give them individual iron pills. What they'll do is they'll usually give them a prenatal vitamin
Yeah, that.
which contains
Yeah.
iron. And they are a big yes, and then then the folic acid, and it's a big part of that as well.
To help that with the extra blood volume, with the body being pregnant and the developing baby inside.
Yeah,
It
so
just kind of boosts that.
and the body will take care of itself, and that supplement is supportive. So
Okay.
because the nutrient needs increase so much during pregnancy. Yeah, they tell people to eat, lots of green leafy vegetables and things like that. They're, if you're not a vegetarian, get your meats in. Your heme is very important. But if you are a vegetarian, you want to just make sure that you're informed. I have friends that are very good vegetarians, and they are informed, and they do very well. It's just important to know where those nutrients are in your food so that you can make sure that you can consume them.
Okay, thank you.
So this is our hemoglobin molecule that we were talking about a little bit later, and then this is the iron containing protein part. So we see that iron molecule there in the middle. Right, so there's 4 segments to it. And then we said that the red blood cells are going to live for about 120 days. And then they're going to be removed from the body. So we see that when they are removed, they removed from circulation by the spleen and the liver. Now, some things can be recycled and reused by the body, and that's kind of cool. So when a child is real young, we whether they're breastfed or formula fed, you'll always see that, especially with formula, that the formula, it'll say contains iron on there. So that is why it is so important. And then breast milk just takes care of itself. And it's, the stores build up and until we hit about puberty, and then we need to really start thinking about, where the sources are coming from. Because we need an increased amount. So this is what we just went over. We just went over what erythropoiesis is. So erythropoiesis is production of red blood cells, and it begins in the red marrow there. And we just talked about erythropoietin. So that is a hormone released by the kidneys in response to hypoxia, okay? And so it's in response to that lowered oxygen concentration, which stimulates the bone marrow to create erythrocytes. All right, and the first one that they're going to see is that reticulocyte, which will enter into circulation and then become an erythrocyte within 1 to 2 days. Okay, so this is a diagram here of just what we went over there. So what are the effects? So I want you to think about something for just a second. if we have, let's say that we had somebody whose kidneys were not functioning properly. All right, so I want you to go ahead and I want you to use your chat box here. All right, and I'm going to be watching. And I want you to think of, just type in what would happen if somebody was on, let's say, dialysis, which is, it's when the kidneys are, not working properly and we have to have a machine that's functioning for them. How would that affect the red blood cells? Like what's going to happen there? Just in your mind from what you've learned. Just type me a really quick message. And if you can't, that's okay. So I have one person. Okay. See if we get a few more people. Something's happening there. Okay. So what happens here is we had, and I would never say names in this. So we have, or I guess you can see the names, huh? Okay. So we have dialysis cleans the blood, which it cleans the red blood cells. Red blood cells would decrease and need to be replaced. It will keep the blood from decaying and keep oxygen within the red blood cells. All right, so he meant dialysis there. And so what actually happens here is I want you to go back for a minute and I want you to think, so we're thinking about dialysis taking care of cleaning the blood. So in this type of situation, we want to say that a person is, the kidneys are failing, all right? So they're not doing the job, and then of course the machine will do the job for it. But in terms of cleaning the blood, remember we said that the kidneys were producing EPO, right? So the EPO is in charge of stimulating the marrow, right, to increase the red blood cells. And what we said was that red blood cells only live, we want to remember that red blood cells, they live about 120 days, all right? So they're going to be, they're going to be dying. So if the kidneys aren't functioning, are they producing these, the EPO? And the answer to that question is during dialysis, they have to administer most of the time EPO. They actually have to give a synthetic version of this. So they have to work to control that red blood cell volume. So that would be the problem in kidney failure, because we need to keep, we follow, what we're following here is the hematocrit, all right? So we want to make sure that we don't give too much. We want it to stay. So remember, we have hematocrit, right? We want it to stay within about 45%. So we don't want it to go too high. If we give too much, that would not be good. And if we don't give enough, that would not be a good thing. Does that make sense? All right. So many things can happen in terms of the kidneys not functioning properly. It would directly affect the red blood cells and many other things that we'll learn about when we get to the urinary chapter. Okay, let's go ahead and move forward a little bit. All right. So now what we're looking at when we look at this here, we're looking at the white blood cells. And the white blood cells are really, really cool. Really cool. All right. So remember where they live. So the white blood cells are going to live within the buffy coat of centrifuge blood. And that means that we have a very low amount, if you think about it, compared to what we see in the rest of the body here. So let's go ahead and think about that for a second. Let me get my thing. So if we're looking at this centrifuge blood again, and we said that we have that buffy coat, very thin layer here. Remember, that's the white blood cells, but also the platelets. So not as much compared to the other formed elements, which we know to be red blood cells. But within this buffy coat of the percentage of white blood cells, we see that we have neutrophils, eosinophils, basophils, lymphocytes, and monocytes. So we have 5 different types of white blood cells. When we think of the white blood cells, we think of our immunity, right? So let's go ahead and let's think about this for a second. I'm going to bring you right down to formed elements here. So the very first one that we see here is we see the neutrophil. And we know that the neutrophil is going to be about 60 to 70% of all the white blood cells. So you want to make sure that you're mindful of that, and that it's not 60 to 70% of the blood of the formed elements, but just of the white blood cell component. So when we think about neutrophils, we think about first what they look like. So these white blood cells here, if I can just kind of draw this out, we know that they have 2 to 5 lobes, all right? So we say that they are polymorphonuclear, right? So neutrophils are polymorphonuclear cells. So they are complete cells here because they have their nucleus and some other important organelles. And we know them to be phagocytes, right? So what that means is it's that they are, eating cells. They're destruction of bacteria. And we know that they fall within this 60 to 70%. They're known as the warriors. So if we have something going on in the body and it's a foreign antigen and the body can't identify it, then these neutrophils are going to go to war for us. So if we have low neutrophils, let's say that we have a number that is lower than normal, right? If we have low neutrophils, we would say that that is neutropenia, right? And we sometimes see that they are, for example, cancer would be, somewhere that we might see neutropenia, low neutrophil values. And what would happen there is that would be where the patient would be immune compromised. So if you are working in the hospital, you might see this on a door for somebody who is immune compromised, neutropenic, and you have to make sure that you have your PPE on and have protective wear, because you yourself could be a threat to this person. Their immune system is not working at the level that it should be working.
I do have a question about these.
Sure.
Could these be classified as T cells?
No, neutrophils are not T cells. They're lymphocytes.
Okay.
So they're they're really like scavengers. Let me see if I can pull this up. So let me attempt to do this here. See if I have we'll go ahead and clear this.
So are T cells like their own thing, or can they fall under one of these types of white blood cells?
They do fall under white blood cells. They are lymphocytes. So we'll be talking.
Oh,
Yes. All right. So
okay.
and so we have B lymphocytes and T lymphocytes. So can you guys see my screen?
Yeah, I can see you typing out neutrophil movie.
Oh, all right. Now this is not in real time. Can you see this?
Is that like sped up or?
Yes.
Okay.
So this is a neutrophil, and then these are bacterium, and then these are crenating red blood cells. So you have some healthy red blood cells in there, and then some ones that are dying off. But let's watch this. Let's watch this neutrophil go to action after this bacterium. So it's a phagocyte.
So it's drawn towards the bacteria.
And you know, foreign substances that the body cannot identify. So do you see how that occurs? So it's going to what? It goes after it, and it's kind of like a scavenger. It's going to pick up those substances. So what it's going to do from there is it's going to break it down, and then it's going to try to use some of the parts if it can. So if you think about that, that was pretty cool, isn't it?
It is.
So it
Yeah.
goes, you know, it's kind of, you know, our warriors. This is, of all the white blood cells, it's 60 to 70%. So if we have an infection or something like that, we want to see in a blood lab, we want to see that these numbers go up a little bit higher so that we know that we are fighting that bacteria, we are fighting that infection. If it stays too low, then we worry if somebody is neutropenic, because we know that their body is not responding as it should. So a bacterial infection might be devastating for somebody that is suffering as opposed to somebody who is not. So that neutrophil is a pretty big deal. So that was phagocytosis, destruction of bacteria. So you learned about like lysosomes and things like that. It's going to engulf that. It's going to digest it and break it down. All right, so this very next one, this next formed element here is a white blood cell known as eosinophils. And these eosinophils are super cool. And if we see a rise in numbers in the eosinophils, it could mean that there's an allergic reaction, or it could mean maybe like a parasitic infection along the way. So it's a couple different things here. So please understand, if you are in blood lab, this is just the very beginning. So we cover it very basically and are in a very basic way. And then if you are in that major, you'll be moving on and learning a lot more. So hopefully we can begin that journey for you here. So if you're looking at the eosinophils here, what we see is we see that the nucleus, and we're always going to be looking at the nucleus to identify these, except for really one that we can't see. It's more like a phone, and you're going to be asked to identify these on your test. So all of the white blood cells, you want to make sure that you know what that nucleus looks like. But one thing that is different about eosinophils is that it is granular, as are the neutrophils, all right? And the granules here are kind of an orangey red. So we have like, you know, like an orange red granules there. So you want to make sure that when you look at this, you can identify it. So we see this, you know, this kind of phone-shaped nucleus, and then these orangey red granules. Okay, so we know that it's only about 2 to 4% of the white blood cells. So we don't always get to see them in lab unless somebody is having some type of a reaction. Right? And the very next one that we have here, oops, let's have to go back, clear that, right? And the very next formed element that we have is our basophils. And our basophils are even less than neutrophils. So they're also very interesting. When we see that, when we look at the basophils, what we see is, it's very hard to see the nucleus. So these cells, we kind of have like an S-shaped nucleus in there, but they're so granule that it's hard to see them. So they are extremely granular. So we usually see that we have two lobes in the nucleus. And that one thing that's very important about the basophils is that they are going to release histamines and heparin. So we have histamine and heparin. So that's going to, one is going to, it's just the blood in terms of, with clotting, and the other one is going to intensify the overall inflammatory response. So when we think about that here, we want to know that these basophils, they're usually very low. They're very hard to find in a lab, and most of the time we do not see them because of their low numbers. But we do look at them in slides and make sure that we understand what their actions are and what their nucleus looks like, as well as the cell themselves. Okay, all right, so the very next one here. Okay. So the next one, we have our cells that do not contain granules. So let's just back up for 1 minute here, right? And let's think about the cells we just went over. So we said we had neutrophils, we have eosinophils, and we had basophils. So each one of these ending in phils, and we say that they are granular. All right, so if they end in phil, it means that they have granules. So neutrophils, eosinophils, and basophils are all granular. But now what we're looking at is the white blood cells, also known as leukocytes, are agranular. So whenever we have a in front of a word, that's going to be not or without. So a means not or without. Right? So if penia meant breathing, right? If penia means breathing, this is just another example, right? Apnea, A in front of the word, apnea would mean not breathing, right? So A means not or without, so agranular means no granules. Okay, so lymphocytes here, and here's your question, Will. When you look at the lymphocytes, we're going to see this nice large nucleus. And sometimes it has a little bit of an indentation to it. And then you see the cytoplasm there is kind of like a purplish color. So we have T lymphocytes and we have B lymphocytes. And what we're going to see is that these cells make up about 20 to 25% of all white blood cells. So we said the nucleus is really large, and what we're going to see is that these B cells are going to develop into what we call plasma cells. But the T cells are going to be important for invading viruses and cancers and things like that. So we know that we have B and T cells. All right, so these are going to develop into the plasma cells, right? And then the T cells are going to be important for viruses and things like that. So if you, when we think about the T cells, we want to think about the viruses and a lot of what is going on right now. So we'll come back to that and we'll talk you know, in our next lecture, a little bit about the role here and how important this is. So for right now, what we're just going to do is we're just going to identify, and then we'll come back and we'll be talking a lot about that within our immunity chapter. Okay, so let's go ahead on to our next slide here, right? So the very last formed element for the white blood cells that is agranular is known as the monocyte. And the monocyte is a cell that is, you know, it kind of looks like a sloppy cell like this. And what happens is it's got a kidney-shaped nucleus, all right? It doesn't have any granules. And we refer to this cell, we say that it's about 3 to 8% of the white blood cells. And we say that it is not only a phagocyte, but it is a macrophage. So it is going to take care of substances within the blood that, you know, are larger and in a big way. So after transforming into a fixed or wandering macrophage, so we have phagocytosis in that process there. Right? So the characteristics are that it's, you know, a large sloppier cell with a kidney-shaped nucleus. Okay, everybody with me so far? Doing okay? Yes, okay. All right, so let's think about this just for another minute. If we have, I can clear this here, so clear. We said that we have our, you know, we have our vacutainer here, right? And we have our red blood cells, which we now know to also be called erythrocytes. And we have our white blood cells here. And the white blood cells we now know to be neutrophils,
Basophils.
right? So we're gonna lay this out in a way that's gonna help you to remember this. All right. So with neutrophils, we have lymphocytes, we have monocytes, we have eosinophils, and we have basophils. All right. So remember, this was 50 to 70%. So this is the largest amount, and then this is less than 1%. So this actually goes in descending order. So we say never let monkeys eat bananas, right? So that would be a way to remember that. You know, our lymphocytes come in around 25%. Our monocytes are anywhere from 1 to 4%. And then these are around 4 to 8% here. And then our eosinophils are about 1 to 4%. So the reference ranges can change from one lab to another. So, and your book has, you know, within these ranges. So the big thing that you understand is that these are our warriors here, these neutrophils. They're the ones that are, you know, they're going to be first and, you know, to respond to non-specific, you know, foreign substances. And as we work our way down, we see that those percentages change. So never let monkeys eat bananas. Neutrophils, lymphocytes, monocytes, eosinophils, and basophils. And remember, they are not the percentage of whole blood, but the percentage of the white blood cells that are going to be contained within this buffy coat. All right. So the next formed element was platelets, right? So then we have platelets that live within this buffy coat. So let's go ahead and we want to think about those because they are the last of our formed elements. And platelets are super, super cool. Sorry to be dorky. Let me go ahead and open this up. All right. So when we think about platelets, right, I'm gonna take you to a slide in just a minute. I want you to think of that slide that I showed you where all of the cells are formed. So we have this, we have this hemocytoblast, right? And this hemocytoblast is going to eventually develop into a megakaryoblast, all right? And then this megakaryoblast is going to develop into what we call a megakaryocyte. All right, I'll take you back to that slide in a few minutes. So this megakaryocyte, one of the cells that is developed within our marrow, all right, what happens is it becomes so large, right, that it starts to break off, all right? So this megakaryocyte, and that's a test question you want to make sure that you're aware of, all right? So it starts to break off. And what we call these little pieces is we call them platelets, right? So these are the platelets that are branching off from this megakaryocyte. And the term is really thrombocyte. And cyte means cell. And it applies to so much of the blood, cyte like leukocyte, erythrocyte, and thrombocyte, meaning platelets. But these aren't actually cells, they're actually like pieces of a cell. So let's think about that for a second. So I'm going to clear this, and I want you to think about that piece of a cell, right? So here we have our, we have our platelet here, right? So this is our platelet. And what we see inside of these platelets is we see different chemicals. It's super, super cool. So we see all these different chemicals. So some of those chemicals that we see are, we have what's called thromboxane A2, all right? We have another one called ADP, adenosine diphosphate, and then we have another one which is serotonin, all right? So I want you to think about that and write that down. So this is very important. So platelets come from that, that we said that large cell, and it's broken off, and this is the megakaryocyte, right? Right, so it comes from this large cell, and then we have the platelets, and then the platelets contain these three very important substances.
What's the top one again?
So thromboxane A2, and we're going to abbreviate like this. So it's called thromboxane A2. All right. So we know that when we think about platelets, we think about clotting cells. So cells that help the body or the blood to clot. And we know that the percent, the amount, we have about 150,000 to about 400,000. So in the blood. So let's think about this. Why do we need our blood to clot? Well, for many different reasons, but one of those reasons, let's say that we think, let's draw out a blood capillary here, all right? And a blood capillary is the thinnest of all the blood vessels, all right? And what it has inside of it is it has this very thin lining known as the endothelium. And we're going to go back and talk about this a little bit more, because it's what we say is that it's semi-permeable. So substances can move in and out of this. But it's got some protective features here. So let's think about this. We have this, this inner part here, kind of like a lipid consistency. And I like to say it's kind of like a Teflon layer. And what this is called prostocyte. cycling. All right, so let's go ahead and put that in on it. So it's like a Teflon layer there. All right, and then on the outer part of this, what we see is we have, you know, this outer layer here, which is a nice layer, right? And we see that we have collagen. All right. So let's say that, you know, we have our blood running, you know, through our blood vessel here, and we know we have our red blood cells and our white blood cells and our plasma, everything that makes up blood. So let's just say that we, you know, one day we have a thorn. We prick our finger with a thorn, so we're looking at this blood capillary, right? So we have this thorn, and what happens is that as soon as this thorn comes along, let me erase that word there, right? As soon as we have this thorn in our finger, it's going to start to throb. So we have a thorn, and what that's going to do is it's going to cause vascular spasm here. So we start to have vascular spasm. So the blood vessel starts to spaz. So that's number one. So let's call that number one, vascular spasm. All right. So number one of what? Number one of what we're going to call hemostasis, which is going to mean the stoppage of blood. So now what we want to do is we want to pull this thorn out. And when I clear this one little bit here, it's gonna cause me to lose some other part of this. All right. So I'm gonna pull this thorn out here. All right. And when I pull that thorn out, what happens, oops, go ahead and put our blood vessel back in place here, right? So what happens is the blood vessel, you know what, I'm gonna go ahead and just do this. Give me 1 minute. I just do this so it makes it a little more pretty and understanding. So you have that in your notes. We'll just make it a little smaller this time. So we have our blood vessel, and then when we pulled that thorn, what happened was it left an open part there. And remember this, you know, now this is kind of disrupted from the vascular spasm and from this being open. And, you know, we have the sticky part out here. This collagen is all been disrupted. All right. And we're going to start to bleed a little bit. So here's our thorn down here, and we've pulled it out, and now we're bleeding. Well, what happens here is that after we have this vascular spasm, then platelets are going to work to aggregate to the area here. And we want to stop the blood from pouring out, right? So we want to stop some of this blood from pouring out. So that would be number two. So we have this platelet plug. So let's go ahead and write that in. So number two is a platelet plug. Okay, so these platelets are going to start to release their chemicals now, right? So now they're starting to release their chemicals as they aggregate. So we have thromboxane A2, we have ADP, and we have serotonin. So I abbreviated that there, all right? So we have ADP and we have serotonin all working together for this platelet plug, right? So what those chemicals are going to do is they are going to further the vascular spasm, but also help platelets to aggregate to the area. So now that we have this, this successful platelet plug here, right? And this vessel has been healed, right? It is not a permanent substance. So it's not permanent. What we need is we need fibrin. So I'm going to think about that for a minute, right? So I need a fibrin mesh here. All right. And the fibrin mesh is actually number 3, which we have is coagulation, right? So then we have these 3 steps to hemostasis. But this isn't so easy. This doesn't come along so easy. So let's go ahead and let's go back and let me share my screen back to our PowerPoint here. We'll go ahead and clear this. All right. And we'll go into we'll go into share this here. And these are our 3 steps. So we have hemostasis, we have vascular spasm, platelet plug, and then blood clotting. Before we go there, you want to remember where those platelets came from. So the platelets are, we see about 150,000 to 400,000. We know that they're cell fragments and that they are going to assist the body in the stoppage of blood. All right. So here we go. Here's our blood vessel, right? And we had our healthy blood vessel. Then we have our injured blood vessel. And if you see here, you see that those chemicals there, we have our collagen fibers on the outside and everything's being disrupted. And then we have they're releasing ADP and serotonin and thromboxane A2. All right. So that's when they're aggregating to the area. And once it's, once we have it, to this area, we can see that we have a little bit of mesh starting to develop there. So once that fibrin mesh is in place, then we have coagulation. So let's go ahead and let's go on to this next slide. So what does the body need in order to form that fibrin? And the answer to that question lives in slide 36 here. All right. So this is important that you understand this. So we have a common pathway here. And I'm not going to take you through all the blood factors, but what I do want you to understand is what happens right here. All right. So let's go ahead and
Miss Marsh, when it comes to somebody being hemophiliac, what is that? Is it a clotting issue? Like you were just describing, they have problems clotting, or is it more of a blood issue?
It's a blood issue.
It is a blood issue, not a clotting issue.
Well, hold your question and then we'll come back to it.
Certainly.
Okay. All right. So if we're looking here, if we are looking at this, we had our blood, our capillary here, all right? And within the capillary, we had the vascular spasm, and then we had the platelet plug. So let's make sure that we can have a good understand. The first thing here, we have vascular spasm, right? And then we have the second thing is that platelets are going to aggregate to the area, the injured areas. They start here. So then we have the platelet plug, right? And then the very next thing is we want to make that permanent here. all right? And with that platelet plug, we want to develop a fibrin mesh. So what we need here is we're first going to need, let me just reduce this a little bit. We're first going to need, can you guys see if I write down here, we need prothrombin, all right? So here is our prothrombin. And what happens is as you see prothrombin, with the help of calcium, we need calcium there, is going to convert to thrombin. So this is inactive, but this is active. So we need it to convert to thrombin. And the way that it does that is it has an activator, all right? So you see this here? We see that we have prothrombin activator here. Right. So this is going to convert, prothrombin is going to convert to thrombin. And the reason is because when it converts, we need that thrombin, this is active, and we now need fibrinogen. Oops. Sorry about that. We now need fibrinogen to convert to fibrin. So fibrinogen needs to convert to fibrin. And the way that it does that is it needs this first. So this here, it needs the thrombin in order to convert. All right. So this is important that we understand this. So we have this prothrombin activator, or the prothrombinase, which is going to convert one to the other. So let me go to the drawing board because I feel like I might be confusing you a little bit here. Go back here and let me clear all drawings. Okay, so when we have that, we had prothrombin, and this is in the clotting part, that needed to be converted to thrombin. And remember, we have that prothrombin activator there. So now that we have thrombin in the blood, another inactive protein, fibrinogen, all right, and this is another factor, needs to convert to fibrin. And the way that it does that is by way of thrombin. So this step needs to occur in order for this to happen. Because we can't necessarily be running around with a bunch of mesh in our body unless we're needing this, right? So we need that fibrin. So what does that do to that clot there? It gives it a mesh-like network, and then we see red blood cells and things like that get caught in that substance. So this is the process. This is coagulation, which requires this fibrin mesh. So prothrombin must convert to thrombin, and then thrombin is needed to convert fibrinogen to fibrin. All right, let's go ahead back to our screen here. Okay, so we see that process there. So these are some of the other factors that we see. We have fibrinogen and we have the prothrombin, and they are, the pathways that they're in are common. But the next thing that we want to consider is what happens when we need to break that clot down. So once the clot forms, it retracts to pull the edges of the damaged blood vessel together. Vitamin K is needed for normal clot formation because it is used in the synthesis of the clotting factors, all right? So these are clotting factors, and we're just covering really these first two. So we know that we need calcium, we need fibrinogen and prothrombin converted into their active forms. And in order for this process to occur, we need vitamin K. So small unwanted clots are usually dissolved by plasmin, an enzyme that is part of that fibrinolytic system. So plasmin, how do we get plasmin? So plasmin is another very interesting substance, because what happens is that in that clot formation, so we had, I'm going to just do this one more time for you. I'm going to do it quick. So we had prothrombin was converted to thrombin. So this is one of those factors by the prothrombin activator. And then we have fibrinogen converted to fibrin by the thrombin itself. So we have the thrombin there. It's an R, right? And then what we have now is we have plasminogen, which is going to be converted to plasmin. So this is really like the what's going to break down the clots. And we need something else to convert this. So what we have is tissue plasminogen activator, and we refer to that as TPA. So tissue plasminogen activator, right? So this is like the clot buster, where it's going to break down those clots. So we have these inactive substances converted into these active substances. That makes sense? Okay. Great. So let's go ahead and clear this here. All right, and go back to our screen share. All right, so when we looked at our class here, we started with what is blood, and the formed elements, and then we looked at all of the formed elements individually. And one thing that we skipped over here is that we failed to recognize how these formed elements are, the white blood cells, are going to move in and out of the blood. So before I end our session tonight, I just want to go over this real quick. So what we see here is we see that capillary that we have drawn so many times. And to be honest with you, once again, we're going to be looking at these capillaries and finding that where they live in each and every body system, because capillaries are where transport is going to happen. So membrane transport that you learned about in Anatomy 1. So you learned about diffusion, and you learned about facilitated diffusion, and all those things, filtration. They are all going to be occurring here within these capillaries. So blood cells can, move in and out of the capillaries if they need to. So when I talk about blood cells, I am referring to the white blood cells. So what we have here is we have this endothelial layer of the blood cell, and what we're seeing in this one picture is we're seeing that we have a white blood cell here, and it's going to kind of roll, it's going to like marginate along the blood cell, and then it's able to squeeze outside of the blood. So when we think about the blood capillaries like this, we think of that a nice endothelial layer, right? And our white blood cells, if they need to move into tissue spaces. So remember, blood and tissues are, the blood and the cells are separate systems here. All right. So these are cells, and more than two or more cells, we make up tissues. So let's say that we have, this blood cell, and it starts to roll along and marginate, and then what happens is it can move into the tissue spaces if it needs to. So when it's leaving through the capillary, that process is known as diapedesis, right? And this process, rolling along the base of the capillary there, is going to be marginization. So let's think about that here. So this process here, this is when it's leaving, it's called diapedesis. So where would it be going? Well, just like we looked in that, that movie, with phagocytosis, it may be moving to clean up debris. So let's say you did prick your finger and, that nail had some substances. So you've opened up that first layer of defense for your body. So if you, let's say you started to have a pussy area, what do you think that would mean? Let's hear from you in chat. So what would it mean if you had if you started to, develop some pus? Well, let's see what you think. So very good. So Corey, that would be infection. So if you had, infection and you had pus, well, the pus is actually dead white blood cells. That means that the bacteria is winning the battle. So they're not, the white blood cells are not working fast enough. So we want to make sure that when we have, infections, that we're seeking help from our physicians if it is a big enough area. So that is, when the body is, certainly if it's a small area, you might be able to use some antibiotic on that and assist your body, and then you can see it, see that helping as well. So the bacteria is pushing the white blood cell out of the infected area. So asking that question, but the bacteria is actually winning the battle. So it's not, the white blood cells are what the pus is. That is the dead white blood cells. So they're losing the battle. So, yep. So we want to, they're moving here. I do want to say that, back to what we said with the characteristics of plasma blood, we said that 91.5% of the plasma is water. So keeping our water levels and keeping ourselves well hydrated is all very important in assisting to the process of the body. So it keeps our solutions moving. If ever you're sick, one of the things that they will tell you is make sure that you're hydrated, because all this metabolic stuff, all the movement of substances within our body, it all happens in water. So water is amazing, and you want to make sure that you stay hydrated at all times, especially when you're not feeling well. All right, everyone. So that's it for tonight, and I took you right up to 7:20. So I did not give you a break, and so there's a nice long session. You all hung in there, and I want to thank you for that. So I'll be posting our lecture for tonight, and I will see you in our next lecture. We do not have class on Monday because it is Labor Day. So what I will be doing is I will be posting for you your exam. So let's go ahead and look at that real quick. In
I
your
have a question real quick.
Sure.
And I may have missed it Monday. Do we not actually have labs with you? We just do it on our own, correct?
So we are doing our, in lecture, we'll go over some of the labs so that everybody is clear, but we don't actually have a full lab session, no.
Okay,
So you'll be
thank you.
working on it. But, I know there's going to be lots of questions as we move through, especially when we get into blood tracing. So we'll be doing some
Yeah.
practicing and things like that on online here.
Another
Thank
question, real quick.
Sure.
How are we gonna have the lab book? I don't know where to get it from.
So you want to buy that from the Owens Campus bookstore. And the reason is because the bookstores are all different in terms of what they're selling. So
No, are they open now? I just don't know.
yeah, no, they are open.
Okay.
Yep.
Thanks.
Hang
Yeah.
on, guys. Let me go into our class here, and
I know that you have to go to the bookstore through the main entrance, must have your ID. They will ask you your name and your 700 code, and they open up at 10 a.m.
Thanks so much.
You're welcome.
thank you for that, Dana. Okay, so let's go ahead and look here at our course. Okay, so when you're navigating the course here, we said that we were in week 1. And what you're going to do here is when you click on week 1, you have all of your information here. But if you look here, let's look at our objectives. So tonight, what we did was we discussed the physical properties of blood, the components of blood, the structure and functions of hemoglobin. So we went right through most of this information. The only thing that we did not cover, which we will do next Wednesday, is the ABO blood groups. So we'll be moving into that and then working on immunity for this. But one thing I'm going to do is I'm going to post some videos for you that's going to get you started on the immunity part, since we don't have class on Monday. So one thing I've done to this point upon request is I've put in a video here for you to watch on innate immunity. And Will, that has some really good information on, it starts with the T cells there at the end, and then here's on the B and T cells, and I'll go ahead and release that. So these are some very good clips, and they're short. They're not too long. If you want to go ahead and get started. So I've watched these over and over and made sure that I'm clear on all the information that it is presenting. Once you've done that, you can create your review questions. And I want to say, class, I do recommend printing your review questions so that, go through, sit down with it with a pencil and see if you can just answer these questions. So, like, what type of tissue is blood? And I want that to come to you very quickly. And I'll be honest, I will be very fair on your exams. Like I said, unless I post something that is going to require you to go outside of the course, I really want all the information that you're learning I want you to be able to find it within what I have posted. So, all these different things that we just, see if you can go through and comfortably answer many of these questions. And you should be able to get down to where it says explain the ABO blood group. So you should be comfortable with all of this by this point. All right. Okay. So, that these 2 videos should get you started for next week. So what I'll do is I'll put these videos probably into an Edpuzzle and look for you to answer questions on those. I will go ahead and release your exam so that if you feel comfortable with your questions and with moving forward, you can do that. But I will recommend that you wait until we have lecture on Wednesday, and then you'll have Thursday through Sunday to take your exam. Makes sense?
Miss Marsh.
Yes.
And another question. So I won't be able to go to a bookstore this week because I'm working till Saturday. So I'm sure they're not open like Sunday. And the Monday is the due date. So I won't be able to get there on Monday. So I don't know what to do. So
Okay, so if you look on at the week 1 for the lab here,
yes.
what you're going to do is you want to complete this lab, so just write it down in a notebook so that you are comfortable with the material, because what the lab book is really just, you're just filling in all the information for that first unit or for the first chapter. So there's a lot of information in there that you're going to need for your practical when we get into the cardiovascular system and so on. But for this very first unit, you should be okay with just writing it down in a notebook and then filling it in later. So, hopefully, because this is giving you want to make sure you can just draw some of the white blood cells, just as I did in the video tonight, so that you're familiar with what the nucleus looks like and working on identification of them. And, we'll always look at a perfect model of a white blood cell. We won't go through all the stages of the white blood cells like you would if you were taking a hematology class. Is that helpful?
Yes, thank you. But it's just, it says assignments due by 9/6/11/59. So
Right. So the only thing that's
I don't want to lose any point from there.
No, the only thing that is due, so this is all fill in. So if you look under your lab, this is the only thing that is due, is the complete. So completed in your lab book. But this blood lab quiz. So this week you have 4 quizzes to take that are all graded. So you have your getting started quiz, which is here under the getting started tab. Then you have your 2 Edpuzzles that you want to complete. All right, so that is in the assignments tab here. So you have these 2 Edpuzzles that I want you to complete, and then you have your blood lab quiz. So I'll make that a little more clear next week. As far as I want you to complete your lab book, but you're not submitting it for a grade this week.
Okay, thank you. I appreciate it.
Just the blood quiz. Okay. All right, everyone. Well, you have a wonderful Labor Day, and I'm going to be online for a little bit longer. I'm going to go ahead and end the meeting, and then I'll come back. I'm going to go get a drink of water. But if you need anything tonight, or if you have any questions, I'll be available. Okay, and then I'll see you next Wednesday. And remember, I do have office hours during the day, and whatever you need. If it doesn't work out, I can schedule individual times.
Thank you.
Thank you.
Yep.
Thank you.
Have a good night.
Thank you.